Dear students, we have learned about the transdermal drug delivery systems. In the previous video, we have learned about definition, benefits, limitations, basic components, and types of transdermal drug delivery systems. In this video, we are going to learn about the manufacturing process and evolution parameters of the transdermal drug delivery systems. In the manufacturing process, first the drug and adhesive solution is prepared by adding the drug material into the adhesive solution. After that, the drug adhesive solution is coated on the backing membrane, and this backing membrane containing drug adhesive solution was dry at particular temperature. After drying, this backing membrane containing the drug adhesive solution was laminated using the release liner, and this laminated film is then cut into suitable size using the die cutters. After that, this suitable size transdermal patch is packed into the packaging material and the final product is ready for the dispatch. These are the few equipments used in the transdermal drug delivery systems in the formulation development department. This is the equipment. In that, the backing membrane is placed and the drug adhesive solution is sprayed on the backing membrane, and the temperature is fixed using this equipment, and the film is dried properly. After drying, this packing membrane containing adhesive solution is laminated using this type of laminators. It is similar like we are making the lamination of our mark sheet. It is similar type of process. After lamination, these are cut into the suitable size by using the die and die cutters. Evaluation or testing of transdermal drug delivery systems. This includes number of tests: physical test, chemical test, cutaneous toxicity, and percutaneous absorption model. Chemical test includes drug content, content uniformity, purity, and the residual solvent is tested in this chemical test of the transdermal drug delivery systems. Physical test includes the release of the drug from the transdermal patches, and the release of the drug is determined by using three different USP apparatus: apparatus 5, pedal over disc, 6, cylinder type of apparatus, 7, reciprocating disc. The physical test also include the test for adhesion, and the adhesion of the film is determined by the rolling ball tag test, prop tag test, peel adhesion test, and the shear strike. Also, the cutaneous toxicity is also determined. That includes the contact dermatitis, growth of microorganism, cytotoxicity, and sensitization study. Another test, that is percutaneous absorption model, that includes in vitro testing and in vivo testing. Now we see each and every test for transdermal drug delivery systems in detail. First, we see the evolution of adhesives. The pressure sensitive adhesives are evaluated by the following properties. Already we have discussed in the previous slide. That includes peel adhesion properties, tag properties, and shear strength properties. First one is peel adhesion properties. Peel adhesion is the force required to remove an adhesive coating from a test substrate. That means the amount of force we are going to apply on the adhesive transdermal patch to remove from a test substrate. Here test substrate means the skin. Whenever we are using the equipment, then the word utilized is test substrate, but when we are applying on the skin and the force required to remove the adhesive patch from the skin, that is known as a peel adhesion force. It is important in transdermal devices because the adhesive should provide adequate contact of the device with the skin and should not damage on the damage the skin on removal. That means it is important because whenever we are applying the transdermal patch on the skin surface, it should be properly adhere on the surface of the skin. And another important thing is, when you remove the patch from the skin, the adhesive solution should not damage the skin on removal. Second one is tech properties. Tech is the ability of a polymer to adhere to a substrate, that means skin, with little contact pressure. So it is a one type of property, that is ability of the polymer that will adhere to the skin surface with a little pressure by using the thumb or finger. It is important in transdermal devices which are applied with finger pressure. Tests for tech include thumb tech test. It is a subjective test in which evaluation is done by pressing the thumb briefly into the adhesive. That means we can place our thumb on the surface of the adhesive and we can evaluate the adhesiveness of the adhesive layer, that is known as a thumb tech test. Quick stick or peel tech test. Here in the figure you can see that this is the blue color transdermal patch. It is stick or adhere on the surface of the stainless steel plate, that is known as a substrate. Here also it is explained, and this is the adhesive layer that will adhere on this surface. And using this equipment, this adhesive tape or patch is pulled away from the substrate, and the force required to break the bond between the adhesive and substrate is measured, and this is known as a quick stick or tech test. During this test, the substrate is pulled at 90 degree at a speed of 12 inch per minute, and the force is recorded as the tech value and is expressed in ounce or grams. Rolling ball tech test. The figure shows that this is the adhesive film, adhesive transdermal patch layer, and this is the ball. Here it is also described, and on this stainless steel plate the adhesive surface is placed, transdermal patch is placed, and the ball is traveled on a surface of the adhesive layer. And here in this test, the distance traveled by the ball on the adhesive film is measured, and this is known as a rolling ball tech test. Next we see about the prop tech test. The equipment shown in the figure used to measure the adhesiveness of the film. Here in this figure you can see here, this is the place where the transdermal patch is placed, and this is the probe. This probe will come and adhere to the surface, and here the force required to pull a probe away from an adhesive at a fixed rate is recorded as a tech properties, and it is expressed in the grams. Third property is shear strength properties. Shear strength is the measurement of the cohesive strength of the polymer. Adequate cohesive strength of a device will mean that the device will not slip on application and will leave no residue on removal. That means how it is important for the transdermal patches, because transdermal patches are applied on the skin surface, and as you know that when we remove the transdermal patch or bandage type of dosage forms, it will create a residue on the surface of the skin. So this shear strength property is very much important to measure that no residue will remain on the surface of the skin, and it will not slip during the application on the skin. So shear strength is determined by measuring the time it takes to pull an adhesive coated tape of a stainless steel plate when a specified weight is hung from the tap. That means this equipment is used to measure the shear strength properties. The blue color tap is a transdermal patches, and this is the stainless steel plate. This stainless steel plate is act as a skin, and this transdermal patch is stick on the surface of this stainless steel plate. After that, the weight of specified amount is hang on these transdermal patches, and the time required to pull off these patches from this stainless steel plate is measured. This is the time scale. From this we can measure the how much time it will remain adhere on the surface of the skin and how much time it requires to remove from the surface of the stainless steel plate. And from this we can check about the shear strength properties of the transdermal patches. In the previous slides, we have discussed about how to evaluate the adhesive property of the transdermal patches. Now we find the in vitro drug release evolution or in vitro permeation studies. This is also known as a diffusion studies, and this is carried out using the France diffusion cells as shown in the figure. The skin permeation of a drug from transdermal systems is evaluated by France diffusion cells with a defined surface area. France diffusion cells have two halves. One is donor compartment and another is receiver compartment. Donor chamber in which the formulation is placed. From this top one is donor compartment, and a receiver chamber that is filled with the release medium and monitor for drug transport. The release medium that is filled in the receiver chamber may be aqueous or hydroalcoholic solution in which the drug is having sufficient solubility and it maintains the sink conditions. Then a suitable size of the heat separated epidermis is cut and mounted placed between the two halves of the France diffusion cell with the stratum corneum side facing the transdermal drug delivery systems. Samples, aliquots of predetermined amount are withdrawn from the receiver chamber at periodic intervals, and the receiver medium is replaced during the transport studies. That means the sufficient amount of sample is collected and the fresh medium is again transferred, fixed amount again transferred into the receiver chamber to maintain the sink condition. All the samples that are collected at particular time intervals are checked, analyzed for drug content using the specified analytical methods like UV or HPLC methods. Drug release profile is checked by using the different USP type apparatus. Mainly USP apparatus 5, 6 and 7 are used. For transdermal drug delivery systems, drug release studies are generally performed using USP type 5 dissolution apparatus. The pedals are used and they are fixed at a height of 2.5 cm from the bottom of the release cage. In each cell, a patch is fixed to the bottom with the adhesive side up. An aqueous solution that provides sink conditions for the release of the drug is used as a release medium. The pedals are rotated at 50 rpm and bath temperature is kept at 32.5 degrees centigrade. Samples, aliquots are withdrawn at intervals at fixed times and replaced with an equal amount of fresh buffer to maintain the sink conditions. The drug content in the samples is analyzed by using suitable analytical methods given in the pharmacopoeia or by UV or HPLC methods. Thank you for patience listening.